Secret the United States Human Biological Experimentation in 1931, Dr. Cornelius Rhoads, under the auspices of the Rockefeller Institute for Medical Investigations, infects human subjects with cancer cells. He later goes on to establish the United States Army Biological Warfare Facilities in Maryland, Utah, and Panama, and is named to the United States Atomic Energy Commission. While there, he begins a series of radiation exposure experiments on American soldiers and civilian hospital patients. In 1932, the Tuskegee syphilis study begins. 200 black men injected and diagnosed with syphilis are never told of their illness, are denied treatment, and instead are used as human guinea pigs, in order to follow the progression and symptoms of the disease. They all subsequently die from syphilis, their families never told that they could have been treated. In 1935, the pellagra incident. After millions of individuals die from pellagra over a span of 2 decades, the United States Public Health Service finally acts to stem the disease. The director of the agency admits it had known for at least 20 years that pellagra is caused by a niacin deficiency, but failed to act, since most of the deaths occurred within poverty-stricken black populations. In 1940, 400 prisoners in Chicago are infected with malaria, in order to study the effects of new and experimental drugs to combat the disease. Nazi doctors later on trial at Nuremberg cite this American study, to defend their own actions during the Holocaust. In 1942, Chemical Warfare Services begins mustard gas experiments on approximately 4,000 servicemen. The experiments continue until 1945 and made use of Seventh-day Adventists, who chose to become human guinea pigs rather than serve on active duty. In 1943, in response to Japan's full-scale germ warfare program, the United States begins research on biological weapons at Fort Detrick, Maryland. In 1944, the United States Navy uses human subjects to test gas masks and clothing. Individuals were locked in a gas chamber and exposed to mustard gas and Lewis-it. In 1945, Project Paperclip is initiated. The United States State Department, Army Intelligence, and the CIA recruit Nazi scientists and offer them immunity and secret identities in exchange for work on top-secret government projects in the United States. In 1945, Program F is implemented by the United States Atomic Energy Commission. This is the most extensive the United States study of the health effects of fluoride, by which was the key chemical component in atomic bomb production, and also is a toxic industrial waste product. One of the most toxic chemicals known to man, fluoride, it is found, causes marked adverse effects to the central nervous system, but much of the information is squelched in the name of national security, because of fear that lawsuits would undermine full-scale production of atomic bombs. In 1946, patients in VA hospitals are used as guinea pigs for medical experiments. In order to allay suspicions, the order is given to change the word experiments to investigations or observations, whenever reporting a medical study performed in one of the nation's veterans' hospitals. In 1946, the United States Public Health Service intentionally infected thousands of Guatemalan citizens with biological agents just to see what the effects would be. This experimentation program continued officially until at least 1948. It is believed that other programs similar to this were, and still are being conducted covertly around the world. In 1947, Colonel E. E. Kirkpatrick of the United States Atomic Energy Commission issues a secret document Document 070750001, January 8, 1947 stating that the agency will begin administering intravenous doses of radioactive substances to human subjects. In 1947, the CIA begins its study of LSD, as a potential weapon for use by American intelligence. Human subjects both civilian and military are used with and without their knowledge. In 1950, Department of Defense begins plans to detonate nuclear weapons in desert areas, and monitor downwind residents for medical problems and mortality rates. In 1950, in an experiment to determine how susceptible an American city would be to biological attack, the United States Navy sprays a cloud of bacteria from ships over San Francisco. Monitoring devices are situated throughout the city in order to test the extent of infection. Many residents become ill with pneumonia-like symptoms. In 1951, Department of Defense begins open-air tests using disease-producing bacteria and viruses. Tests last through 1969, and there is concern that people in the surrounding areas have been exposed. In 1953, the United States military releases clouds of zinc-cadmium sulfide gas over Winnipeg, St. Louis, Minneapolis, Fort Wayne, the Monocacy River Valley in Maryland, and Leesburg, Virginia. Their intent is to determine how efficiently they could disperse chemical agents. In 1953, joint Army, Navy and CIA experiments are conducted in which tens of thousands of people in New York and San Francisco are exposed to the airborne germs Serratia marcescens and Bacillus galargii. In 1953, CIA initiates Project MKUltra. This is an ongoing research program designed to produce and test drugs and biological agents that would be used for mind control and behavior modification. 6 of the sub-projects involved testing the agents on unwitting human beings. In 1955, the CIA, in an experiment to test its ability to infect human populations with biological agents, releases a bacterium withdrawn from the Army's biological warfare arsenal over Tampa Bay, Florida. In 1955, Army Chemical Corps continues LSD research, studying its potential use as a chemical incapacitating agent. More than 1,000 Americans participate in the tests, which continued until 1958. In 1956, the United States military releases mosquitoes infected with yellow fever over Savannah, Georgia and Avon Park, Florida. Following each test, Army agents posing as public health officials test victims for effects. In 1958, LSD is tested on 95 volunteers at the Army's Chemical Warfare Laboratories for its effect on intelligence. In 1960, the Army Assistant Chief of Staff for Intelligence authorizes field testing of LSD in Europe and the Far East. Testing of the European population is codenamed Project Third Chance. Testing of the Asian population is codenamed Project Derby Hat. In 1965, Project CIA and Department of Defense begin Project MK Search, a program to develop a capability to manipulate human behavior through the use of mind-altering drugs. In 1965, prisoners at the Holmesburg State Prison in Philadelphia are subjected to dioxin, the highly toxic chemical component of Agent Orange used in Vietnam. The men are later studied for development of cancer, which indicates that Agent Orange had been a suspected carcinogen all along. In 1966, CIA initiates Project MK Often, a program to test the toxicological effects of certain drugs on humans and animals. In 1966, the United States Army dispenses Bacillus subtilis variant Niger throughout the New York City subway system. More than 1 million civilians are exposed when Army scientists drop light bulbs filled with the bacteria onto ventilation grates. In 1967, CIA and Department of Defense implement Project MK Naomi, successor to MK Ultra and designed to maintain, stockpile and test biological and chemical weapons. In 1968, CIA experiments with the possibility of poisoning drinking water by injecting chemicals into the water supply of the FDA in Washington, D.C. In 1969, Dr. Robert McMahon of the Department of Defense requests from Congress $10 million to develop, within 5 to 10 years, a synthetic biological agent to which no natural immunity exists. In 1970, funding for the synthetic biological agent is obtained under H.R. 15090. The project, under the supervision of the CIA, is carried out by the Special Operations Division at Fort Detrick, the Army's top-secret biological weapons facility. Speculation is raised that molecular biology techniques are used to produce AIDS like retroviruses. In 1970, United States intensifies its development of ethnic weapons military review, November, 1970, designed to selectively target and eliminate specific ethnic groups, who are susceptible due to genetic differences and variations in DNA. In 1975, the virus section of Fort Detrick's Center for Biological Warfare Research is renamed the Frederick Cancer Research Facilities and placed under the supervision of the National Cancer Institute. It is here that a special virus cancer program is initiated by the United States Navy, purportedly to develop cancer-causing viruses. It is also here that retrovirologists isolate a virus to which no immunity exists. It is later named HTLV human T-cell leukemia virus. In 1977, Senate hearings on health and scientific research confirmed that 239 populated areas had been contaminated with biological agents between 1949 and 1969. Some of the areas included San Francisco, Washington, D.C., Key West, Panama City, Minneapolis, and St. Louis. In 1978, experimental hepatitis B vaccine trials, conducted by the CDC, begin in New York, Los Angeles and San Francisco. Ads for research subjects specifically ask for promiscuous homosexual men. In 1981, first cases of AIDS are confirmed in homosexual men in New York, Los Angeles and San Francisco, triggering speculation that AIDS may have been introduced via the hepatitis B vaccine in 1985, according to the journal Science 227 173-177. HTLV and VISNA, a fatal sheep virus, are very similar, indicating a close taxonomic and evolutionary relationship. In 1986, according to the Proceedings of the National Academy of Sciences 83-4007-4011, HIV and VISNA are highly similar and share all structural elements, except for small segment, which is nearly identical to HTLV. This leads to speculation that HTLV and VISNA may have been linked to produce a new retrovirus, to which no natural immunity exists. In 1986, a report to Congress reveals that the United States government's current generation of biological agents includes modified viruses, naturally occurring toxins, and agents that are altered through genetic engineering to change immunological character and prevent treatment by all existing vaccines. In 1987, Department of Defense admits that, despite a treaty banning research and development of biological agents, it continues to operate research facilities at 127 facilities and universities around the nation. In 1990, more than 1,500 six-month-old Black and Hispanic babies in Los Angeles are given an experimental measles vaccine that had never been licensed for use in the United States. CDC later admits that parents were never informed that the vaccine being injected to their children was experimental. In 1994, with a technique called gene tracking, Dr. Garth Nicholson at the MD Anderson Cancer Center in Houston, TX discovers that many returning Desert Storm veterans are infected with an altered strain of Mycoplasma incognitus, a microbe commonly used in the production of biological weapons. Incorporated into its molecular structure is 40% of the HIV protein coat, indicating that it had been man-made. In 1994, Senator John D. Rockefeller issues a report revealing that for at least 50 years the Department of Defense has used hundreds of thousands of military personnel in human experiments and for intentional exposure to dangerous substances. Materials included mustard and nerve gas, ionizing radiation, psychochemicals, biological and chemical agents, hallucinogens, experimental technology, and drugs used during the Gulf War. In 1995, the United States government admits that it had offered Japanese war criminals and scientists, who had performed human medical experiments, salaries and immunity from prosecution in exchange for data on biological warfare research. In 1995, Dr. Garth Nicholson uncovers evidence that the biological agents used during the Gulf War had been manufactured in Houston, Texas and Boca Raton, Florida and tested on prisoners in the Texas Department of Corrections. In 1996, Department of Defense admits that Desert Storm soldiers were exposed to chemical agents. In 1997, 88 members of Congress sign a letter demanding an investigation into bioweapons use and Gulf War syndrome. In 2009, Vietnam veterans bring a lawsuit against the CIA for implanting remote control devices in their brains, and using them for a myriad of biological and chemical warfare tests in addition to the still classified and yet to be revealed torturous human experimentation programs that will be brought to light in the coming months and years. This is just a short list of the last century of evil covert human experimentation programs. If we are to see an end to this evil, we must share this knowledge with as many as we can, and help raise awareness of these issues, so that these mistakes will not be repeated in the future. For those who do not remember the past, are doomed to repeat it.
We are anonymous. We are legion. We do not forgive. We do not forget. Expect us.